Hey class, so in the second video on cell respiration, I'm going to cover mitochondrial structure and how that's related to the link reaction, Krebs cycle, oxidative phosphorylation, and electrons transport chain. I know you're terribly excited, so I'm going to jump in. Okay, 813 states that draw and label a diagram showing the structure of a mitochondria as seen in electron micrographs. So if we were to draw this, I would draw a mitochondria, and then it would be like kind of like a mouth, you know. But basically, what this is saying is that mitochondria have two lipid bilayers, which is very unique. It's the first structure that we've talked about that has two lipid bilayers. There's the outer membrane, and there's the inner one, and the inner one has these folds in it. And the folds are called cristae. And the cristae are providing increased surface area, so things can pass in and out really easily. Remember, when you have increased surface area on the outside of an object, you can get things out and get things in much more easily. An electron micrograph looks like this. It looks somewhat similar, but you should be able to tell the difference between a idealized drawing and the actual thing. It has these lines in the middle. So, okay. What have we covered so far? So far in the first video, we covered this much. That you take a 6-carbon molecule and you make 2 3-carbon molecules, and you get 2 net ATP produced, right? You put 2 in and you get 4 out. Okay. After that, we have another series of reactions. So the first one is this thing called a link reaction, and that's explained here. So let me read these two assessment statements. It says, explain that during aerobic cell respiration, pyruvate can be broken down into carbon dioxide, water with a large yield of ATP. So this presentation, this video, is going to explain to you how we get this large yield of ATP. The next assessment statement says, explain aerobic respiration, the link reaction, the Krebs cycle, the role of NADH and H+, the electron transport chain, and the role of oxygen. So, the link reaction. In the link reaction, you're adding this coenzyme A, to your pyruvate molecule. But first, you're going to oxidize, right? You're going to oxidize. That means remove a carbon and add oxygen to it. And you're going to remove one of them. So you're going to have a 2-carbon molecule, and you're going to add this enzyme CoA, this coenzyme factor. NAD becomes NADH. And you get this product which enters the Krebs cycle. So you see here, pyruvic acid letting the CO2 out, making acetyl-CoA. Then the CoA gets shut off and you get into this cycle. So you have this molecule OA, which you don't need to know the names of. You take your 2-carbon and you add it to the 4-carbon to make 6. If CO2 gets pumped out, you have a 5-carbon molecule. Another CO2 comes out, you get a 4-carbon molecule. A series of reactions, you still have 4, you still have 4, you still have 4, and then it cycles through again. You add another acetyl-CoA, and you get a 6, and then a 5, and then a 4, and then it goes back into the cycle again and again and again for every acetyl-CoA that comes through. And as this cycles, you produce 1, 2, 3 NADHs, 1 FADH2, and 1 ATP. Now, this ATP isn't that significant in number, but it's this NADHs and FADH2s that are really powerful in creating more ATP. The next assessment statement says, explain oxidative phosphorylation in terms of Kimmy osmosis. So, you notice that this inside space of the inner membrane is called the matrix. And the Krebs cycle goes on inside the matrix. And these NADHs and FADH2s are used to pump H pluses into this intermembrane space, which I'll show you on this next figure. So let's take a look here on the left. You notice that the NADH becomes NAD plus, and when that electron gets released, it pushes H plus into the intermembrane space. Notice there's a lot more H pluses in this intermembrane space, so the pH is really low. It's very acidic. A little further down the line, FADH2 is also losing its electrons, and it gets into this. So what's happening is the electrons are going down this chain of proteins and other factors. You don't need to know the names of them. There's a figure like this one on the right in your book. And what it's showing is the electrons are jumping from one complex to another. And the reason they're doing that is because it's slightly lower energy than the previous one. When it does this in a stepwise fashion, it's doing it gradually, and not much energy is being lost in the form of heat. So from FMN to FES to Q to C to cytochrome B to this other FES to cytochrome C1 and so on. Again, you don't need to know the names. The idea is that the electrons are being transferred slowly as if they're going down a hill towards less energy, and eventually that electron gets transferred to oxygen to make and hydrogen to make water. When you create an imbalance between the outside of that inner membrane in the intermembrane space and the matrix, the H pluses want to flow back towards the matrix. And when they do that, they flow through this molecule called ATP synthase. It's a protein that is embedded inside the inner mitochondrial membrane. And when it flows through, this ATP synthase synthesizes ATP from ADP. And this is the big generator of ATP in cellular respiration. If you don't have oxygen at the end to collect the electrons, it's like breaking the circuit. So I don't know if you remember from physics, when you create a circuit, it has to be completed. If you break the circuit, the process doesn't occur at all. So if you don't have oxygen, there's no place to collect the electrons. This whole process stops. So that's the importance of oxygen. So in the last assessment statement here, it says, explain the relationship between the structure of the mitochondria and its function. So to explain this, you see you have the outer membrane, and the intermembrane space in between the inner membrane and the outer membrane is where the hydrogens collect. So that means the pH is going to be very low, right? H pluses are high, pH is low. Because the cristae are folded like this, there's a lot of surface area for this process of pumping hydrogens across the membrane into the intermembrane space. And then when you create this imbalance, you can create ATPs when the H plus flows back into the matrix, which is this space inside the double membrane. And there's a good table on page 226 that summarizes the 5 parts to the mitochondria, which you should look at. So with that, I hope you write some questions down, and I will review this with you in class. Thanks.